Welcome to today's lecture on cells and organs of the immune system. So before we get started, I want to make sure that you remember to follow along with your Janeway textbook, as I'll be referencing figures that you will be able to access in that textbook. As we begin, I just want you guys to remember that immunology is incredibly complex, and for today's lecture, I want you to focus in general on the concepts as opposed to the details that are contained within the lectures. And we're going to be learning about the organs and cells that are contained within the immune system, and we're only going to be introducing them today, and we'll be looking at them in much greater detail in coming lectures. Today, our main learning objectives will be to understand the major differences between myeloid and lymphoid cells, as well as being able to describe the lineages of myeloid and lymphoid cells. We'll be following this discussion with major differences of primary and periphery lymphoid organs, and then we'll be talking about the general properties of peripheral lymphoid organs. Now, the immune system is spread throughout our entire bodies. As you can see, there are organs contained in almost every part of our body. However, the organs of the immune system can be subdivided into four major categories. First, sites of immune cell development. Second, systems by which immune cells are trafficked throughout the body. Third, sites at which the immune cells are exposed to antigens, and sites at which the adaptive immune system is activated. Now, we'll be going through these four major general sites and looking at them in further detail. The first portion that we'll be talking about are the sites of immune cell development. Now, again, in this slide, I'm just going to go over the major terminology that we'll be going through, and I'll define it throughout the lecture. The two sites of immune cell development are the thymus, which is located just behind the heart, and the bone marrow. We'll then be looking at systems of immune system trafficking, which includes the bloodstream and the lymphatic system. We'll then be discussing sites at which antigens are exposed to the immune system, and these include tissues, blood, and mucosal surfaces. And finally, we'll look at sites at which the adaptive immune system is activated by the innate immune system, a concept you were introduced to in the previous lecture, but we'll be going through again today. And these sites are lymph nodes, spleen, and mucosal surfaces. Now that we have a very brief overview, let's jump in at the very start, which would be the sites of immune cell development. Now, let's start off with the bone marrow. So the bone marrow is a very critical organ. It's actually the semi-solid tissue that's found in the spongy or cancellous portions of bones. And it's also the site where all cellular components of blood are generated. And it's the site where most cells of the immune system arise. And it's also the site where many cells of the immune system will develop and mature. So now I'll take you through a very general overview of immune cell development, and subsequently we'll delve into more specific details. So in the bone marrow, most cellular components of the blood, including the cells of the immune system, arise from pluripotent hematopoietic stem cells in the bone marrow, seen here. From the bone marrow, the pluripotent hematopoietic stem cells develops into the common lymphoid progenitor cell and the common myeloid progenitor cell. Both of these components subsequently give rise to a variety of different components of immune cells. From the bone marrow, a majority of immune cells will migrate into the blood, and subsequently they'll migrate into either lymph nodes or tissues, a variety of other places within the body, and potentially some of them will further differentiate. Now, if we step back and look at the schematic as a whole, we'll be going through almost every portion of these developmental stages today in a very broad overview, and subsequently more maturation processes will be gone through in further lectures. So I find that the best place to start in terms of development tends to be at the beginning, so let's start at the very top of this hierarchy with our pluripotent hematopoietic stem cell in the bone marrow. So in order to define pluripotent hematopoietic stem cells, it's sometimes easier to break this word down into its components. So stem cells are cells that have the capacity to produce both a self-renewing stem cell as well as a second cell that has a capacity to differentiate into a large variety of specialized cells. Now, pluripotent stem cells have a little bit more specific of a definition, such that they are stem cells whose progeny, so for example, that second cell, can form multiple specialized cells. So the cells that a pluripotent stem cell's progeny can make are more defined relative to a general stem cell. Now, pluripotent hematopoietic stem cells are stem cells that give rise to various different types of blood cells, including immune cells, all of which we'll be going through today. So let's go back to our very large chart, which I realize seems intimidating, but let's break it down and take one step down that hierarchy from pluripotent hematopoietic stem cells down to the two major progenitors that are formed from the pluripotent hematopoietic stem cell, which are the common myeloid progenitor cell and the common lymphoid progenitor cell. So let's start with the common myeloid progenitor cell. So the common myeloid progenitor cell, again, is differentiated from the pluripotent hematopoietic stem cell. And this myeloid progenitor cell in itself is also a progenitor for other cells. And it can give rise to granulocytes, which are white blood cells with multi-lobe nuclei and cytoplasmic granules, which you can see here. Mast cells, which are very large cells that have many granules, as you can see here. Phagocytes, which are cells that are capable of engulfing and absorbing bacteria and other small cells and particles within their environment, as well as megakaryocytes and erythrocyte progenitor cells. So these are all the various different types of cells that our myeloid progenitor cells can give rise to. So now let's dive a little bit deeper and understand all the various functions that these cells have. So first, let's talk about the megakaryocytes and erythrocytes. If we can start with megakaryocytes and erythrocytes, I don't want to go too much into detail about these cells, but it's important to understand that these also arise in the bone marrow because they're both components of cells that are components of blood. Megakaryocytes are very large cells, as you can see here, that are responsible for the production of platelets, which are really necessary in blood clotting, which is important in wound healing. And erythrocytes, which are red blood cells, which are here, are anucleated blood cells that transport oxygen and carbon dioxide to and from tissues. Now again, we won't be going into these cells much further, but it's important to note that they are also a progeny of the common myeloid progenitor cell. As we shift gears into a different portion of the progeny of the common myeloid progenitor cells, these are our granulocytes. So granulocytes in general, which I have outlined here, are innate immune cells named for densely staining granules inside their cytoplasm. You can even see here in their cartoon depictions that they have these little spots within their cytoplasm that represent the granules. These cells also have very oddly shaped nuclei in that they are kind of much elongated and kind of curved relative to the traditional, more spherical nuclei. And again, you can see this graphically represented here. In addition, these cells are very, very short-lived. They survive only for a couple days. And there are three major types of these that are specifically distinguished by different staining patterns of their granules, and they serve distinct functions. Now again, I want to emphasize that we'll be going through very broad properties of these three different cell types, and in future lectures on the innate immune system, we'll be delving more into their specific functions. Our first cell that we're going to briefly talk about is our neutrophil. So a neutrophil is a phagocytic innate immune cell. So this means that it can uptake various things from its environment by engulfing them. It's also one of the most numerous and most important cells of our innate immune systems. They take up a variety of microorganisms by phagocytosis and destroy them by these intracellular granules that we see here. So they also have a variety of different bactericidal mechanisms that again we'll delve into in our next lecture on the innate immune system. The two other components of the granulocyte family are eosinophils and basophils. So these are much less abundant within our immune systems relative to neutrophils. However, they also contain the granules containing variety of bactericidal and antimicrobial enzymes and toxic proteins that neutrophils have. However, they specifically function mostly against parasites and within allergic reactions. And again, we'll be going through more details of these cells in future lectures. However, it's important to note that very general properties of these granulocytes. Again, granulocytes are composed of neutrophils, eosinophils, and basophils, and in general they have the property that they have granules within their cytoplasm that contain antimicrobial and bactericidal enzymes, and they have these elongated, oddly shaped nuclei. Now, another cell type that has parallel properties to the granulocyte group are the mast cells. So this is another subset of innate immune cells that derive from the common myeloid progenitor cell, and they also have granules within them. So these are very, very large cells, and they actually begin their development within the bone marrow. However, they leave as immature precursors, and they actually mature within peripheral tissues. Now, as you can see, compared to the pictures of our granulocytes, there are much more, the granules contained within mast cells are larger and much more dense, as you can see by the deep purple staining here. These granules contain histamine and other active agents that work to protect our bodies against invading pathogens, predominantly parasitic worms. So now that we've talked a lot now about granule-containing cells, and very general properties of these is that they're innate immune cells that contain granules that contain bioactive enzymes that function to protect our bodies against invading pathogens. Now, as we move on to another set of cells that also derive from the common myeloid progenitor cells, we'll now be shifting our focus to phagocytes. So the general properties of these cells that are highlighted here in red are these cells are capable of engulfing and absorbing bacteria and other small cells and particles from their environment. So again, this is one property of the previously discussed subset of cells called neutrophils. However, because the neutrophils' main function isn't phagocytosis, they're not included within the phagocyte family. Now let's start with monocytes. So monocytes develop in the bone marrow, and just like our mast cells, they're not mature when they leave the bone marrow. They actually head out from the bloodstream where they can encounter parasites and pathogens and such, and then they're actually the largest type of white blood cell that are phagocytic, and they usually circulate within the blood for about 2 to 3 days before they migrate into tissues. And as I mentioned briefly before, these cells are not fully developed when they leave the bone marrow. Once they enter into tissues, these monocytes will actually differentiate into two other subsets of phagocytic cells called macrophages, or they'll develop into dendritic cells. So macrophages in general have the property of being phagocytic, and they're also really important in various bactericidal mechanisms. They're resident in most tissues, so almost every one of our tissues in our bodies have what's called resident macrophages. So what's interesting about these macrophages is that the tissue-resident macrophages are actually macrophages that have not derived from blood marrow-derived monocytes. They actually arise during embryonic development. So even when we're born, we have a variety of different macrophages which are resident to specific tissues. However, macrophages that develop from bone marrow-derived monocytes are also incredibly important in our immune responses. So macrophages are also really critical in a process called antigen presentation. So antigen presentation, briefly, is a process by which cells, predominantly phagocytes, will uptake various proteins and molecules and other cell types from the environment, bring these molecules in or cell types in, break them down, and present a portion of those proteins on their surface. And these presented proteins or presented antigens are really critical in activating the adaptive immune system, which you heard briefly about in the previous lecture. And I'll go into a little bit more detail in a few slides. The other cell type that's really important, that's also a phagocyte, is called a dendritic cell. So dendritic cells are also phagocytes. However, they're very visually distinct from macrophages, as you can see, because they have these very long, elaborate processes that are very similar to the dendrites of nerve cells. So immature dendritic cells migrate through the bloodstream from the bone marrow to enter various tissues. Now, at these peripheral sites, the dendritic cells will use their dendrites to sense the environment as well as uptake various particles from their environment. They're also very, very critical in antigen presentation and are usually thought to be the bridge between innate and adaptive immunity. So let's take a step back now that we've gone through a majority of the myeloid lineage. So again, just to review, myeloid cells are those that arise from the common myeloid progenitor cells. These cells are part of the innate immune system, which means that they function to identify and eliminate pathogens without specificity. If you think back to some of the properties that we've considered of these cells, they tend to be properties that would affect various pathogens under various immune stress situations. Some of the subsections of cells included within the myeloid lineage are the granulocytes, which contain granules with active biomarkers that kill or inhibit the growth of pathogens, and phagocytes, which engulf pathogens. So now that we've gone through a large variety of the common myeloid lineage, let's shift gears to the other cell derived from pluripotent hematopoietic stem cells, the common myeloid progenitor cell. So if we look at this lineage of cells, you can also see that the common myeloid progenitor cell is also itself not fully differentiated. Now, the common lymphoid progenitor cell is the precursor of a variety of cells that are in the lymphoid lineage. These include the natural killer cells, which have the ability to recognize and kill tumor cells or cells that are infected with intracellular pathogens, innate lymphoid cells, which are a very newly described group of cells that have properties of both innate and adaptive immune cells, as well as the lymphocytes, which are composed of the B and T cells, which are adaptive immune cells. And these express distinct types of antigen receptors and have little functional activity until they encounter a specific antigen that interacts with the cell-specific antigen receptor on their cell surface. So before we jump into lymphocytes, let's go through the natural killer and ILC cells. So our natural killer cells are very large lymphocyte-like cells that have very distinct granular cytoplasms, and they were actually recognized because they are able to directly kill tumor cells or virally infected cells. So these cells will actually release lytic granules that kills virus-infected cells, and they play a very important role in the early immune response, the innate immune response. In addition, the other cell that is derived from the common lymphoid progenitor cell are the innate lymphoid cells. So these ILCs reside in peripheral tissues where they function as sources of mediators of inflammatory responses. And they also have overlapping properties with different classes of lymphocytes. So these are a very newly described subset of cells, which are a very active area of current immunology research. And so we won't be going into their functions in too much detail today. So as you can tell, we've kind of gone through NK cells and ILCs and all of our myeloid lineage cells, and these are cells that are components of the innate immune system. Now, before we delve into the specific properties of lymphocytes or T and B cells, I want to go over and review a little bit of the difference between innate and adaptive immunity. Now, if we look at the immune response in general, if plotted against time and the total immune response, After an exposure to a specific pathogen, there is a not pathogen-specific innate immune response that is launched against that pathogen. However, after a few days, there is the induction of a highly specific adaptive immune response. Now, this portion of the immune response, the adaptive immune response, is carried out predominantly by two major cell types, our B cells and our T cells, and these are also called our lymphocytes. So a few major lymphocyte properties are that their response takes a few days to initiate, and the major reason for this is because these cells are highly specific for particular epitopes on antigens. And what do I mean by this? So if we look at the receptors of T and B cells, represented here by these Y-shaped molecules that are red and blue, these receptors recognize specific portions of antigen proteins. So for example, here, the red receptor specifically recognizes this red portion of the antigen, whereas this blue receptor recognizes this blue portion of the antigen. These sites that are recognized by these receptors are called epitopes. So each individual cell of the adaptive immune system recognizes a distinct epitope of antigens, such that the response is highly targeted, highly specific. However, it does take a few days to initiate and to stimulate. Now, another very general property of lymphocytes is the clonal selection hypothesis of lymphocytes, and this is an idea of how they develop and how they mature within the periphery. First, each lymphocyte bears a single type of receptor with unique specificity after it derives from the common lymphoid progenitor cell seen here. So if we start with the common lymphoid progenitor cell, the lymphocytes during development gain different specificities to various proteins. Afterwards, obviously we don't want our immune system acting against our own proteins that we have in our bodies. And so lymphocytes that have receptors that are specific for self-antigens, represented here by these red squares, these cells will be eliminated. And so once your lymphoid cells have been eliminated from self-reacting cells, what we're left with is a group of lymphocytes that have specificities for foreign molecules. Now, when there is an interaction between said foreign molecule or foreign antigen and the lymphocyte receptor for which it's specific for, this will lead to the activation of that specific lymphocyte. So, for example, in this example, the yellow cell that is specific for the circular antigen here will be activated, and then this activated lymphocyte will subsequently proliferate and differentiate into what's called an effector cell. So this is a highly active functional cell which will bear receptors of identical specificity to those of the parental cell.